What is HIV and AIDS? HIV is a virus that spreads through bodily fluids that affect certain cells of the immune system, called CD4 cells. What are CD4 cells? Well, CD4 is a glycoprotein found on the surface of immune cells. There are many kinds of CD4 cells, such as T helper cells, monocytes, macrophages, and dendritic cells, all of which are essential to fight off infections. At this point, the person infected with the HIV virus has the disease AIDS. How does HIV infect the body? The best way to prevent yourself from being infected by HIV is to be informed about its modes of transmission and avoid situations where transmission is highly likely. HIV is transmitted through unprotected sexual contact, shared needles, accidents through occupational exposure, and more. Unfortunately, HIV can be transmitted through blood transfusion and through childbirth also. How exactly does HIV enter a healthy white blood cell? HIV relies on the presence of two receptors on the cell. The primary receptor is the CD4 receptor, and the secondary receptor is the co-receptor, CCR5 or CXCR5. Once the virus is attached to both of these receptors, the cell and the viral envelope collide, and the viral capsid enters the host cell. The host cell's digestive enzymes disintegrate the viral capsid so that the single-stranded viral RNA and the three viral enzymes are exposed to the host cell. The virus contains two identical copies of single-stranded RNA and three enzymes: reverse transcriptase, protease, and integrase. Reverse transcriptase immediately begins to make a DNA copy of the viral RNA genome while degrading the RNA strand. Reverse transcriptase then creates a complementary DNA strand. Then the ends of the double-stranded DNA bond non-covalently. The double-stranded circular DNA then enters the host cell's nucleus and is incorporated into the host cell's DNA using the enzyme integrase. With host cell activation, RNA may be synthesized by DNA polymerase, yielding messenger RNA and viral genome RNA. Once mRNA and the viral genome RNA leave the nucleus, mRNA is translated into the viral enzymes, reverse transcriptase, integrase, protease, and it's also translated into some structural proteins. Some of the structural proteins translated from mRNA are cleaved by the enzyme protease. The structural proteins surround the newly made enzymes and the viral enzymes to form a capsid. The virion is then released by budding. Protease digests the polyprotein chain into its component parts. This allows those parts to come together to form the mature structures that make the final capsid. Now, the mature virion is free to infect other immune cells, destroying the immune system as it progresses.